een eer om hier vandaag bij jullie te zijn. Dank u wel om hier te zijn. Ook dank aan de organisatoren van deze wetenschappelijke sessie. En vooral Dr. Anne Jolie, met wie ik een goede uitwisseling heb opgebouwd. Mijn excuses, ik ga presenteren in het Engels, omdat mijn Nederlands onvoldoende is. So I will start with what is stem cells. So it's a new approach. We create a new concept which can be now considered as something more feasible for new therapies, and that's what I will try to explain to you now. But there is still a lot of work to do. So, slide. Of course, we are talking about ALS, and before that, and before talking about stem cells, I will talk about the one who, in fact, the first one who described correctly the disease. And the next slide is Jean-Martin Charcot, and as you see, it was more than 150 years ago. He was the first French neurologist from, he worked at the hospital La Salpêtrière in Paris, and he was the first to establish a neurological service in Europe. And he was an excellent teacher also, and this is why I show you the picture of him and many students. And so he was the first one to really describe correctly the symptoms of many diseases, and among them ALS. And we had many students such as Gilles de la Tourette. Gilles de la Tourette discovered another neurodegenerative disease called Gilles de la Tourette syndrome. We also had other big names like students like Babinski, and now it's still used by the neurologists as a test, Babinski test, and many others. So I want just to let you know that it was more than 150 years ago, and it was still the middle age for many diseases. But I will move to stem cells, and for that we have a little cartoon or animation to show you or to try to explain easily what a stem cell is, and maybe I will translate this for you.
Wat is een stamcel? Deze stamcel heet Guy, en het is een cel die overal in het lichaam komt, maar waarvan de functie, wat die cel precies doet, nog niet bepaald is. Het heet stamcel omdat elke cel in ons lichaam afstamt van dit type cel. Vandaar. Een stamcel moet u vergelijken met een theaterspeler, een toneelspeler die wacht, rustig wacht, achter de gordijnen, tot er een signaal komt dat hem vertelt welke rol hij moet spelen. Dus deze acteur heeft heel veel mogelijkheden, kan heel veel rol spelen, maar hij wacht. En terwijl hij wacht, verdeelt hij zich, vermenigvuldigt hij zich, zoals u ziet, en wacht dus op een bevel, op een signaal om zich verder te specialiseren. Hier ziet u de stemcel en die gaat zich heel langzaam specialiseren. Wij noemen dat differentiëren in één bepaalde celsoort en één bepaald celtype. Hij wacht op signalen. De signalen gaan genen activeren en die genen gaan hem vertellen welke eiwitten er aangemaakt worden, want die eiwitten zijn nodig om een bepaalde functie uit te voeren. De stemcel begint zich te specialiseren, wordt anders zoals u ziet, dankzij de signalen en blijft zich vermenigvuldigen. Dus die specialisatie, differentiatie is heel variabel en het is ongelooflijk wat een stamcel allemaal kan in de evolutie naar een meer gespecialiseerde, differentieerde cel. Zoals je op de volgende print ziet, kan de stamcel eigenlijk kiezen welke functie hij later zal krijgen. En wij zijn uiteraard geïnteresseerd in de nummer 4, dat is de zenuwcel in het Engels neuron. Dus als wij vragen aan de stamcel, en kan je nu ons eens tonen hoe je een neuron wordt, dan zal hij zeggen, ja, ik ben een neuron. Een neuron dat dient om signalen door te geven, bijvoorbeeld om een beweging in gang te zetten. Een stamcel is er al voor wij geboren worden en de meeste zijn ook aanwezig en gaan soms hier. Wij zullen zien nu hoe die stamcel een zenuwcel wordt en u zal heel typisch de structuur herkennen. Als u van boven naar beneden kijkt, dan ziet u bovenaan een stam, een neuron, met een celkern en aan de bovenkant vertrekkende wortels, dat zijn de dendrieten. En het signaal wordt doorgegeven langs die uitloper, dat noemen we bij een axon, die beschermd is met myeline. U moet zich dus voorstellen dat een signaal, bijvoorbeeld een beweging, lijkt op een doorgeven van een signaal via een telefoon. Dat is kort geschetst hoe een stamcel kan evolueren tot een zenuwcel. Kan u mij achteraan horen? Ja. Nu is het beter. Ja, goed. Dan geef ik graag terug het woord aan professor Pochet.
Thank you, Anne, for this description of what a stem cell is. Now we will see how we could cope and try to see how the progress were made with those stem cells. So, but the main problem is to transfer this to human, because embryonic stem cells, as you can realize, comes from embryo. And so the main source being human embryo create, of course, a major ethical problem. And this is why it was there are many regulation and research where in some countries like Italy stopped on that topic. But there is also a way to reach this, or to get more stem cells, and that's what I'm going to try to explain to you now. And this makes mostly the topic of the Nobel Prize, who was allocated last year to two main researchers from two different generations. It's John Gurdon from Cambridge and Shinya Yamanaka. And they were able to show that they can use mature cells, differentiated cells, and reprogram them into stem cells, so to go backwards. And this is a major discovery, because it was always think that you cannot go back. Once you go from stem cells to differentiated cells, you cannot go back. But they showed and demonstrated that it was possible. So next slide. So this is exactly what I just explained. Maybe you can read it. And so John Gurdon, what he did, so John Gurdon made experiment in 62. This is more than 45 years ago. And what he could show is that by transferring a nucleus, so the nucleus is the part of the cell which contains all the genetic information, by transferring the nucleus of adult cells into stems, into embryonic cells, he could generate a new animal. This means that indeed the material contained in the nucleus was able to regenerate and give a stem cells. So next slide. So he used the tap tool is kicker, and he used stem cells from the kidney. He deleted the nucleus and injected, transferred the nucleus, only the nucleus, from a differentiated cell into this cells empty of nucleus. And then there was a mysterious steps in which the nucleus became, thanks to the environment, thanks to the cytoplasm, the content of the cytoplasm, come back to the stem cell status, and then from this stem cells it could generate a full tattoo. This was the basis of one of the cloning of animals, and they could do it in different animals. So this was in the 62. It was only on nucleus, but it was never. So the difficulty was from cell, do the same things within the whole cell. And I just want to make a small parenthèse here about John Gurdon. We used to know John Gurdon at UNB. Next slide. Because we collaborated with him, and it was a time I was doing my not thesis, but my license work with Gérard Marnet and John Gurdon, and they used, in fact, his frog and the oocyte. This is just to illustrate that science is a difficult and complex matter, and you can use a lot of techniques before. And this was in the 71. So next slide. So the second owner of this Nobel Prize was Yamanaka. Of course, another generation, 45 years later, it could be in fact shown that the whole cell could be, major whole cells can be reprogrammed into stem cells. And this was a major step forward. So, next slide. So this is a principle. He used major stem cells. The cells is a long cell, so the nucleus called a fibroblast, a cell from the skin. He injected into the cell some DNA with a virus, so it would transfer some genes. Only four genes. We have transcription genes we could order the different genes to be expressed, and those cells comes from normal mice, the skin from the mice, and then after transferring the genes, you could generate stem cells, which is pluripotent cells, so which have all the capacity to express all the genes. And from these cells you could generate new mice. So this was the main issue, the main discovery. So we may ask, why did it take 40 years to go from the nucleus to the whole cell? And this is the next slide. Well, this illustrates that we need more and more tools and new techniques and new discovery to reach such level. And this was maybe we discovered new tools, mostly the restriction enzymes, and this was enzyme discovered in bacteria, and researchers discovered later that those enzymes are very useful for the human DNA, and also the whole genome, which is a sequence of DNA, was the biggest to sequence the genome was the biggest experiment of the last century, and it was promoted or driven by Watson also a Nobel Prize, who discovered in '53 that DNA was a double helix. And Watson, thanks to his reputation, could generate necessary funds from the National Institute of Health to install the most important project in terms of money, the genome project. And so they could do the whole genome, human genome project. And this was also an important tool and need information to do this. And also how to transfer genes, this means to make a gene outside the cells into the nucleus and make it express. So all this was needed to reach the experiments of Yamanaka, and it took more than 40 years. So this is just to illustrate that basic research is indeed needed and it takes time. This is very complicated to explain to politicians, because politicians, like Winston Churchill used to say, for politicians 8 days is eternity. So this also illustrates the difficulty to make simple for basic research. Next slide. So the principle was now established that from a cell taken from a human body, example from your skin, we could generate single cell, cultivate cells, make the transfer of genes, generate stem cells, and from those stem cells, depending on the condition of the culture and different factors, those stem cells could then give either neurons or muscle cells or hepatocytes, cells from the liver. And you can then study individually and in vitro culture those cells. Next slide. So the concept is now established that from a cell, a mature and differentiated human cell, you can isolate them, you can transform them into induced pluripotent cells, stem cells. Then you can differentiate them into different types, such as neuronal, preliminary or potent cells. It's an intermediate state of neuronal cells. And then from this cluster of cells, you can go a step further and differentiate them into different types of neuronal cells, such as neurons. And from these neurons, you can cultivate them. They can establish, like it was shown in the cartoon, some contact. And from this, you have an in vitro system from a human cells. This means that if you take cells from a patient who has an ALS disease, for instance, you can generate iPS cells and finally some neurons and look in vitro which drugs can modify their behavior, or what is the, compared to normal cells, what is the difference in electrophysiology or morphology or different parameters, and try different drugs to try to come back to the normal situation. And this is illustrated in the next one. So next one. Yes, it's this first step, followed by the second step is a resume. You can have iPS cells from patients suffering of ALS, familial or sporadic, and then grow neurons, then look at the specific phenotype. The phenotype is how the cell looks, and after that, with this, try different drugs to modify the phenotypes and going back to normal situation. So this is the concept. So now come back to ALS, and what we did in our work is that to use this transplantation of stem cells for cell therapy. Of course, we haven't reached this level yet, but we are progressing. Next step. So we can, what we have used an animal model for ALS. And those are rats which overexpress a human gene coding for a protein called SOD1. When it is mutated, this protein gives the disease. So this is a familial form of the disease. And those rats, when they overexpress the human genes, have the characteristic symptoms of the disease, such as fasciculation, loss of weight, and palsy of the limbs. So we use this model. We have an ALS model, an environment model, and what we do is to use human iPS. We generate human normal iPS, transplanted them in those rats, the spinal cord of the rats, and look what happens to those iPS. If they die, if they differentiate, if they transform into tumor, that was we were looking at. So next step. So what we use is, well, we have the iPS from different human sources, control from sources. One patient suffering from a familial form with a mutation of the GDP protein, and also from ALS, but we limited ourselves at this work to normal iPS cells first. So this is the most complicated slide. I'm not going to go into all the details. It just shows you that we could use from human fibroblasts, differentiated them into iPS or stem cells. But as you can see, this is a very long process. It took 3 months to go from this step to this step. And then we had to isolate the clones, different type of cells, and characterize each clones to be sure that they are really iPS. This means that they can be differentiated into all kinds of cells. And this is here, but I'm not going into the details. So we had those iPS cells, and we transplanted them, next slide, into rats. So for this experiment, we used nearly 50 rats, and we injected them about 200,000 stem cells in a volume of 2 microliters into the spinal cord of the rat. And we looked what happens to those cells. So those human cells were labeled, we could follow them. They have a marker, so we could see them by fluorescence. And the next slide. So the protocol was the following. So this is the base of the rat. We injected something called D0. But before that, D-5, we gave them some cyclosporine, which is an immunosuppressant, because the cells in human are different from the rat, and there can be some rejection, some intolerance, of course. So cyclosporine allows them to be adopted. So, and then we looked at different days. So we isolate rats and kill rats at different days and looked what happened to the cells we have injected. So to show you those, next slide. So those rats, at 90 days of life, they show motor neurons loss. They have started the sign of disease, which is fasciculation and loss of weight. And at 150 days, they have palsy of their limb. And to make a long story short, we looked at different days, but at 60 days we could see those kind of cells, which are human cells, because they are fluorescent in green. This is a marker, and they have the morphology of motor neurons, and they were located in the spinal cord of the rat. So this means they can survive, and they can also be differentiated in motor neurons. So, what our experiment did not show yet is that it was not for cell therapy. It was too early. So if we wanted to look at cell therapy, this means how the rats behave after this transplantation. It's another next step forward. We need to make more injections because the number of cells you inject once, many dies, and we are convinced they need to have more cells, so they need more injections. And also we haven't looked at the parameters of who can control the progress or the non-progress of the disease, which is some electrophysiology, to look at the fasciculation, to look at the weight, and also to test the motor neuron capacity by the test called Quadrout. So, yes, also there was one aspect we have to be very careful, is that because you transfer some genes which they can express different genes which may be responsible for tumor progression, such as nesting. We haven't seen in the rats any tumors progressing, but of course it was only 60 days, which is too early. But this is one of the aspects we have to control, that indeed after gene transfer they don't develop tumors. So this is what I just said, I think. So, and what should be done? What should be done, of course, is to look more carefully at the donor cells, to improve the reprogramming technology, because it's very long. There are more and more progress in that sense. Many experiments are done, and there are now papers showing that they can move from mature cells directly into neurons without passing through the embryonic steps, but it's still very experimental. So last but not least, all those research could be done, need to be funded, and because ALS is a rare disease, funding is more and more difficult. So we have the support of different patient associations or patient family, and here are the three family or patient associations who helped us. It was the family Rosenstein from Eupen, who had familial disease, and they collect different funds. And Mrs. Smith, her husband died from ALS, and she also organized, with the help of the Lions Club in Mons, a big concert which allowed to get 24,000 for the lab. And also, this is the Paul cartoon, and the son, this is the brother and son. They were all. She died a month ago, unfortunately, and the son organized a running of 80 kilometers, twice a marathon of Brussels. He could raise some money for the lab. I want to thank them especially, and I want to thank you for your attention. Thank you very much.
Zijn er mensen die iets willen drinken, dat er platzaken en spuitvaten voorzien staan, dan ben ik daar heel in de zaal.
Het onderwerp voor stamcellen is een moeilijk onderwerp. Ik wou u vragen welke vragen u heeft voor professor Pochet. Wie durft de eerste vraag te stellen? Ja, meneer.
Hetgeen die de professor uitgelegd heeft, lijkt voor ons veelbelovend. Nu voor een aantal ziekten waar men nog altijd aan het zoeken is om te kunnen genezen. Ik denk aan het laatste wat ik hoorde was een verband met suikerziekte, waar men ook nog niet helemaal rond is. Durft men toch een prognose maken. Zeg ja, wij denken met de huidige stand van onderzoek dat wij binnen 10 jaar een oplossing gaan hebben. Ik heb de indruk dat men nu al zou kunnen zeggen, we gaan het volgen.
So the presentation was so promising, but there are other diseases for which we will need to oppose. And the question is, for diabetes, they make a prognosis. It will take maybe 10 years to have a solution in human. Could you give us an idea how long for ALS it would take in terms of years to also have this kind of pregnancy problems?
Yeah, well, it's an interesting question. Of course, it depends on many factors. It depends on how fast the government wants to promote this kind of research. This means they have to. It's a political question, not only a scientific question, because as you heard, there are ethical issue. So this has also to be solved fast by lawyers, and as you know, lawyers are not very fast. And if you put enough amount of funds and stimulate and make a lot of calls for research, it will promote and it will fasten the system. But I think I would say that another 10 years will be needed.
Ik vertaal snel, dus het is een interessante vraag die niet alleen een wetenschappelijke vraag is, maar ook een politieke, omdat dus die stamcellen ook ethische vragen oproepen. En het is eigenlijk aan iedereen om voldoende rugbaarheid te geven en fondsen te geven, zodanig dat politiekers bereid zijn om dat op een agenda te zetten, om dat sneller te laten vooruitgaan. Er komen bij die ethische kwesties ook vragen voor advocaten en dat werkt nog altijd langzaam. Maar als professor Pochet een prognose zou moeten doen voor de toepassing van stamcellen in ALS, dan denkt hij aan een termijn van ongeveer 10 jaar.
Waar wij gewacht worden op de microfoon alsjeblieft, want ik heb hier achteraan in de zaal vragen.
Ik zal de vraag zo in het vraag stellen, maar het vertaal misschien iets gemakkelijker. In de Verenigde Staten is men nu bezig met een debat om ervoor te zorgen dat patiënten eigenlijk op vrijwillige basis kunnen deelnemen aan wetenschappelijk onderzoek, aan dus de bepaalde experimentele therapieën, om dus de ontwikkeling daaraan te versnellen. Bestaat eigenlijk dat debat ook in België. Donc, aux États-Unis, il existe maintenant un débat entre les patients et les billets en ce qui concerne les tests sur des humains, donc des volontaires, pour accélérer un peu le développement des thérapies. Est-ce qu'il y a un débat pareil? Est-ce que ça existe aussi en Belgique? Est-ce que c'est quelque chose qu'on peut envisager? Parce que je pense qu'il y a pas mal de gens qui voudraient essayer des choses. Si ça n'aide pas, et ça n'aide pas, tant pis, mais...
Je vais répondre en français alors.
Oui.
Or in English?
English, please.
Okay. Yes, the point is very important, and indeed, with such disease for which there is nothing, no cure, no therapy yet, it is possible to generate through the ethical committee through the ethical committee or the agreement of the ethical committee of the hospital, it is possible to generate clinical trials rather fast. That's one aspect. And the other aspect raised is the influence of the patient association becomes more and more important. And if patient association say what they should do, it's more and more what is called lobbying, general term, to influence the authority to allow such practice which doesn't follow strictly the rules of all the clinical trials. But it is thanks to the push of the patient association that this can be fastened, increased and implemented.
Een manier om het proces te versnellen is om te werken via de ethische commissies die in de ziekenhuizen verantwoordelijk zijn voor de goedkeuring. Als er klinische studies gebeuren met experimentele behandelingen, dus de ethische commissies benaderen. En anderzijds is er belangrijk lobbywerk voor de patiëntenorganisaties. Ja, zij kunnen, zoals de laatste jaren is aangetoond, ook een push geven om het onderzoek iets sneller en misschien via minder strenge regels, iets minder strenge regels te laten doen.
Wie had nog een vraagje? Ik neem aan, al hetgeen dat we juist gezien hebben, ik bedoel van voor die muizen, dat er nog nooit op mensen zijn gebeurd. Dus het is misschien hoopvol, maar ik denk dat het voor ons hier allemaal een beetje te laat is. Dat we gaan nog eens 20 jaar moeten wachten op die zo'n dag.
We have seen experiments on rats and mouses, but 20 years for us is much too long. I don't know if such tests are also done in humans. Maybe I can just begin and tell you that at the end of this afternoon I will present to you 3 studies that are going on, 3 of the many that are going on in patients, this time with stem cells, and then we will come back on how long it will take until more patients will be able to get these stem cells. But there are studies, these actually now in patients too. Ik wil daarop inpikken en zeggen dat er vandaag de dag al studies lopen met stamcellen bij patiënten met ALS. En dat is waarover ik aan het einde van deze middag ook een presentatie zal geven. Maar de grote vraag blijft, hoe lang zal dat nog duren? Want voor ons is 10 jaar veel te lang.